Okay, thank you very much, Kata. It was a very nice presentation and a lovely discussion. So we have to move on. Then the next presenter will be Alexandra Demcsák from UCLA. Heterozygous SPINK1 deficiency promotes progressive pancreatitis in mice. Alexandra.
Thank you for the kind introduction. And before I would go on to my talk, please allow me to congratulate Peter Hegyi for this milestone. It's such an honor for me to present at the 10th anniversary of the Hungarian Pancreatic Study Group. So the SPINK1, or by its legacy name SPINK3, is a small trypsin inhibitor protein, which is synthesized in acinar cells, and then it's stored in zymogen granules, and then it's secreted into the pancreatic juice. Its major role is to prevent trypsinogen activation to trypsin within the pancreas, and it can also inhibit active trypsin in the pancreas. So based on the currently available data, loss of function mutations of human SPINK1, which are most of the time are in the heterozygous forms in patients, they increase the risk for pancreatitis. And the hypothesis behind this is that the reduced level of SPINK1 will inhibit less amount of trypsin in the pancreas, and that will increase the risk for pancreatitis. However, this was never thoroughly investigated in animal models, and the currently available mouse models are giving contradictory results, like about this hypothesis, what is really going on in the background. So there can be two situations. One, when SPINK1 is in a reduced state, or it can be increased in the animals. So let me start with what is happening when the SPINK1 is reduced in the mice. So when it is in a homozygous SPINK1 deletion state, then the pancreas of these mice has progressively disappeared after birth. These mice fail to gain weight, and then the mice died at around 2 weeks of age spontaneously. On the other hand, when it is in a heterozygous form, they develop completely normally, and their pancreas is showing a completely normal structure. So that's why previous groups, and even in our study, we only investigated in these heterozygous mice since the homozygous mutation is legal. So when this research group investigated what is happening when they induced acute pancreatitis in these heterozygous knockout mice by sertraline injections, they did not find any difference compared to the control mice. So on the other hand, when SPINK1 was elevated in mice, in case of these transgenic mice which are overexpressing SPINK1, they could show that there is protection against sertraline-induced both acute and chronic pancreatitis. So the problem is coming from these two facts, that when you reduce the SPINK1 level, it has no effect on the disease severity. On the other hand, when you increase the level of SPINK1, it will be protective against pancreatitis. And none of the previous research groups have investigated if this could be due to a trypsin-dependent mechanism or not. So our aims in the current study was to investigate the effects of heterozygous SPINK1 deletion on acute pancreatitis, chronic pancreatitis, and intrapancreatic protease activation. For this, we generated a new mouse model with CRISPR-Cas9 mediated genome editing that resulted in the complete deletion of SPINK1. Then we induced acute pancreatitis by 10 times sertraline injection. We also measured the intrapancreatic protease activation after one sertraline injection. We also followed the progression of the disease after an acute episode, and we also investigated the disease course of spontaneous pancreatitis in the T7B23A SPINK1 knockout cross-strain. So before I would go into the details of these experiments, let me just summarize how are these heterozygous knockout mutant mice. So they, as previous groups have found it, they develop completely normally. And by real-time PCR, we could show that the SPINK1 mRNA level is about 50% compared to the control mice. And this also showed on the protein level, verified by Western blot analysis, that the SPINK1 level, even on the protein level, it is decreased in these SPINK1 knockout heterozygous animals. And when we look at their H&E stained histology sections, I'm not sure how much it's visible from the back, but on the left side, you can see a 6-month-old control mouse and a black 6 mouse pancreas. And on the right side, it's a SPINK1 knockout strain. And they were basically identical. So they didn't show any spontaneous phenotype by aging. So then we induced acute pancreatitis in these mice with 10 times sertraline injection. So they got 10 sertraline injections hourly, and they were euthanized 1 hour after that. We measured their pancreas weight and the plasma amylase levels. And you can see that in the SPINK1 knockout heterozygous mice, after the sertraline injection, we can see a significant increase compared to the saline-treated mice, both in case of the pancreas weight and the plasma amylase level. However, it did not show any difference compared to the sertraline-treated control mice for both parameters. So we also evaluated their age and stain sections. We evaluated 3 parameters: the edema, inflammatory cell infiltration, and the rate of necrosis. All of these 3 parameters were comparable between the 2 strains. And from these sections, I would like to highlight the bottom 2 because these are the highest magnifications. So again, on the left side, you can see a sertraline-treated black 6 mouse's pancreas. And on the right side, this is from a SPINK1 knockout mouse. And even on the picture, so these are the ones which we evaluated, but even on the picture, you can see that there is no difference between the 2 strains. So we went further, and then we wanted to measure the intrapancreatic protease activation in this strain. For this, the animals received only 1 sertraline injection, and then they were euthanized 30 minutes after that. We measured both intrapancreatic trypsin and chymotrypsin activity. And again, similarly to the previous results, we found that in the sertraline-treated mutant mice, there is an elevated trypsin and chymotrypsin level. However, again, it did not show any difference, significant difference compared to the trypsin and chymotrypsin activity in the sertraline-treated control mice. So taken together, these 2 experiments, we can see that the reduced SPINK1 level has no effect on acute pancreatitis. And basically, these results are in correlation with what the previous groups have found. So we went further, and we wanted to see how is the disease progressing after a sustained stimulation. So for this, the animals received 8 times injections on sertraline injections on 2 consecutive days, and they were euthanized on the 5th day. And remarkably, the pancreas weight of both sertraline-treated groups, so in the control mice and both in the heterozygous mice, the pancreas weight was decreased. However, as it is marked with this red square, it was more prominent in case of the mutant mice. And when we looked at their histology sections, so this is from a sertraline-treated black 6 mouse, and these 2 are from the heterozygous animals. So you can see that there is more atrophy in the mutant mice. There is a lack of histology restitution with variable degree of acinar cell damage. And the intact acinar cells are in these mutant mice were, like the average was around 60%, but as I said, they showed a really variable picture. So to conclude this experiment, we could see that the SPINK1, the reduced SPINK1 level actually has an effect after chronic stimulation. So it has an effect on the disease course. So while we can see healing in the black 6 animals, which has barely any damage or no damage at all, the disease is further progressing in case of the SPINK1 knockout heterozygous mice. So I know that Kata already mentioned this mouse strain, but so we started to cross these animals with the T7D23A mice. So this mutation is in the T7 trypsinogenes activation peptide, and it was investigated by Andrea Ghez, and it was recently published. So when she investigated in vitro what is happening, she could see that this mutation is causing a 50 times faster autoactivation. So when this mutation was introduced in the animals, that caused a spontaneous acute pancreatitis, which then further progressed to chronic pancreatitis. And the age of onset was around 4 and 5 weeks of age, which is showed by the elevated pancreas weight here. So we crossed these single mutant T7D23A mice with the SPINK1 knockout heterozygous strain. And this is what we found at 3 weeks of age. So this is the pancreas, the result of the pancreas weight, and the cross mutant mice, so which are carrying both mutations, they had a significantly decreased pancreas weight compared to the control mice and also the single mutant counterparts. And when we look at their histology pictures, you can see that in the cross mutant strain, there were 2 kinds of pictures when there is no healthy pancreas structure at all, or almost completely damaged pancreas. On the other hand, at the same age, you can see either normal or acute pancreatitis in the single mutant T7D23A mice. So we can see that when you introduce this mutation into this T7D23A mouse strain, the disease onset has shifted to an earlier time point. So to summarize our data, the reduced SPINK1 level has no effect on cerulean-induced acute pancreatitis and intrapancreatic protease activation. And based on our data, the SPINK1 animals are more prone to progress to chronic pancreatitis after an acute episode. And the SPINK1 deficiency increases the severity of spontaneous pancreatitis in the T7D23A mice. So in conclusion, our data is supporting the notion that the heterozygous SPINK1 deficiency promotes progressive pancreatitis in mice in a trypsin-dependent manner. And thank you for your kind attention.
Questions already there?
Beautiful work. There's been some reports that the SPINK1 molecule, or the pancreatic secretory trypsin inhibitor, has other functions besides inhibiting trypsin. And I'm wondering if you're seeing two unrelated facts. One of them is more trypsin injury driven, and the other one is a failure to heal or to modify the immune system. And if there's a way of teasing out whether or not this SPINK1 actually has multiple mechanisms, and you're focusing on the right one.
I completely agree with that, that they can have other functions too. So it has been already written down in the literature that they might be behaving like the EGF1, and there are suggestions that yes, so the lack of SPINK1 can lead to less of histological restitution in these organs, or sorry, in the pancreas in this case. But yeah, that certainly can be. We didn't go into that so deeply yet.
It's pretty striking, and they don't seem to match quite as well as you would have anticipated, at least.
Yeah, it was really surprising to see that SPINK1 knockout mutation in a heterozygous form on its own cannot cause anything, right? Like you can see that it doesn't cause spontaneous disease, but once you put it together with this really robust mouse model, and I already tried it with another trypsinogen mutant, it really enhances the pathogenesis, and it shows a remarkable phenotype at a really early age.
Very nice work. Just one question about the homozygous mice. So what's the cause of that?
I actually really don't know. So that was, I tried creating homozygous mice, but I had the same result as that Japanese group. So basically when they are born, they're completely identical to their litter mates, but once they start to grow, they're just really small. They have this runty feature, they have less of hair, and the pancreas nest is completely empty. So I really don't know. It can be because maybe the pancreas is that much digested that they cannot grow because they cannot digest their food. That's maybe an idea, but I don't know the cause of death in these animals.
How does the pancreas look?
There is no pancreas.
No pancreas.
Yeah, so this Japanese group wrote down that the pancreas is developing completely normally until I think around 14 or 15 days in embryonic state, and then it starts to disappear. And for example, the duodenum starts to extend and spleen gets smaller. So I really don't know what is the cause of that.
It may correlate with trypsin expression you get.
Yes, we thought about that too, yeah.
So congratulations to your talk. So as far as I understood in the last 20 years, that when trypsin is activated, after there are two ways how this can disappear, either inhibited by SPINK1 or degraded by CTRC. If I look at my research, the cellular one, the calcium channels, ATP, it's so connected, these kind of very similar mechanisms. So if one mechanism is over or maybe overstimulated, downstimulated, the other channels, transporters, switching and taking. So I am almost sure that in the trypsin field, this kind of co-working between the mechanism of trypsin activation and degradations are playing a role. But as far as I hear plenty of presentations from you or from Miklós's group, I very rarely see that you not only pick up and investigating one of the inhibitors, but also investigating the others. So my question is that whether did you have any attempt to look at, for example, in these animal models, how CTRC work? Either these are overexpressed, not overexpressed. So how the other trypsin mechanisms work?
I think that's a really great idea. We haven't looked at it from that point of view yet, but I think it's a great suggestion. Thank you.
Thank you very much. Any more questions? No? Thank you very much, Alexandra.